Okay, so I will start and I'll change the slide. Well, I think I'll change the slide. Okay. So I want to try to emphasize concepts, but still I have to talk a little bit about detail, but I'll try to make it so that the detail doesn't really obscure the basic concepts. And I think this is kind of a summary. How do proteins make everything work? How do cells talk to the world outside of their cell wall? What is specificity and how is it achieved in nature? And how is the diversity that's crucial to our immune system achieved in nature? And what does activation mean in an immune system? And there's some terms, I think I'll try to just go over these as they go along, but I think everybody knows what an antibody is. An antigen is the molecule that the antibody is specific for. And if I say something like that and it's not clear, please interrupt me. So an antibody finds an antigen, or the antigen finds the antibody. And the rest of this stuff I think we'll come to as things go along. So, to kind of be journalistic about it, DNA and RNA get all the hype, I think, and they deserve it, and they put the players on the field, but proteins are the players. They do everything, virtually everything, every process that the cell is involved in, and I listed some examples here. So I think a concept is to understand how the structure of proteins really gives them the kind of variability that they have that enables them to do what they can do. And so I'm going to talk about these things. So don't get overwhelmed by this, but this rectangle or square, whatever it is, has chemical representations of chemical structures of the 20 amino acids that are the building blocks that make up all the proteins in our body. And I just want to, I put a legend here if you really want to look into this further, but the point is to point out a couple things that are on the left here. If you look at each one of these has this structure, and the way these are oriented, it's always on the bottom. It has this structure that's identical in every single amino acid, and this is the chemical structure that allows it to hook up with another amino acid. The NH2 hooks up with the COOH on the adjacent amino acid. And by contrast, every single one of these amino acids has a different group that sticks out from this structure. So when these amino acids are put together in a chain, there are going to be all these different groups that have different chemical properties sticking out from the surface of that chain, in effect. Is that notion clear? So, so these are the levels of, there are 4 levels of structure for a protein. This square up here in the left-hand corner shows the backbone chain being formed by bonds between those structures that I showed you. And they all form a sequence of amino acids that ultimately is determined by the genetic code, which I'll show you in a minute, where 3 nucleotide bases code for one particular amino acid. So if you think about it for a minute, there are almost, the amino peptide chain can be, a protein chain can be 100 or more amino acids. So there are many potential variations in how these can line up. And then the protein chain is going to fold, and in folding it creates some secondary structures. This is a helix, and this is what's called a beta sheet or a pleated sheet. And then it folds up into a more complete structure, and these are different ways to represent protein structures. This one kind of represents the secondary structures with helixes and things like that. And then there's yet a fourth level where a folded protein structure in its mature form can associate with yet another protein molecule and form complexes. So the point is really that there are a lot of ways that you can have variability in proteins. And the next slide is really just a lot of words that says that. And so by folding up into a three-dimensional structure with these groups sticking out, you can form sites that are charged sites, sites that are non-charged, what we call hydrophobic, and you can create any number of, any kind of topology, and that's what enables the protein to carry out the function that it's going to carry out. And these are examples of some of the stuff that sticks out from the different amino acids, and you can actually attach another molecule, like a sugar molecule, covalently to one of these sites. I should say, I want to define what a covalent bond is. I'm assuming everybody doesn't know exactly what a covalent bond is. So if you go back to atoms, an atom consists of a nucleus, which is positively charged, and an electron cloud that surrounds it. And when two atoms share their electron cloud, that's a covalent bond. And that's the most stable kind of chemical bond. And I think I'll mention covalent bonds, but is that idea clear? Okay. So even though protein is folded up into a particular structure, it still has a little bit of flexibility and wiggle room, and that'll become important in a way I'll show you. And then a protein may fold up into a shape that gives it a site with some specificity. And by specificity, I mean that there's an affinity for a molecular structure that is for that molecule, but not for something that's closely related, but not identical to that molecule. And that specificity would occur at what is often called an active site. And protein molecule may have one or more active sites. And so in the lab, you can copy proteins, you can copy DNA and RNA with enzymes. And that's what PCR does. And it is a process that was discovered in 1983, and it's opened incredible avenues of research and application. You can't do that with a protein molecule. There are no known enzymes that can just take a protein molecule separate from the cell and make a copy of it. And if you think about the different structures of the amino acids, that's part of the reason. And this is not essential, but I thought it was interesting. These are the forces that maintain the protein structure. And so I'm going to describe for a second each one. These are water molecules, and this is an oxygen atom in the water molecule of H2O. And an oxygen atom has an electron cloud that makes it slightly polarized negatively. And this is a hydrogen in another water molecule that its electron cloud is polarized positively. So there is a weak force between them called a hydrogen bond. And this is water actually has a structure, and there's all kinds of literature on the structure of water. But these bonds are not weak, but they're sufficient that they can, in combination with other forces, be influential. They're very important in the double helix, for example, in DNA. And then there are some, in that collection of amino acids, some of them were positively charged on the outside and some of them were negatively charged. So attractions across that kind of sharing could form a bond. This is an example of a covalent bond, a representation of it, where these two sulfur atoms are actually bonded covalently to each other. These others, this is the strongest bond, and this is probably the only kind of bond, and it's less common. And then there's something that may be less evident, and I think it's interesting, which is called a hydrophobic bond. And it's the concept that you see when you put oil in water and it all layers out on the top, and you can stir it up, but if you leave it sit, it will still all settle out on the top. And that's because thermodynamically, water molecules would much rather be around other water molecules and not around things that don't have an oxygen or hydrogen or something polarized sticking out on the outside. So some of the amino acids that were in that framework that I showed you are what are called hydrophobic, and the attraction of hydrophobic moieties in avoiding water around it is another force that can act not only to hold the protein structure together, but also to bind something to the active site. And this is just to illustrate what I mean by specificity. This is kind of representation of the surface of the protein, and this is another representation. We used to talk about it as a lock and key, but that's pretty rigid. But this is just molecular structure that's complementary to the site that's been created on the protein that is specific for it. And we say those two molecules have a binding affinity, and the protein surface region is specific for the molecular structure. We have different names for the things that bind to an active site. One is a general term is a ligand. If it's an enzyme, it's called a substrate. If it's an antibody, it's called an antigen. Okay, so are we okay so far? Okay, and this is, I think, interesting. So I said that these things are among the other things that can stick out from the surface of a protein. And these are things that can actually have another molecule of a different kind, like a sugar, attached to them. And the cell does this sometimes because it labels proteins that are supposed to go certain places in the cell or are being labeled for destruction or whatever. And it's the active site around these particular moieties when it occurs that tells the enzymes in the cell to put a sugar on it. So in HIV, it turns out that the virus takes advantage of this by evolving sequences that attract sugar attachment to the outside. And this is a cartoon of the surface molecule of HIV, and the sugars that get attached to that surface actually form a shield. And it's one of the reasons, not the only reason, it's one of the reasons why it's very difficult to actually create a vaccine for HIV, because the antibodies can't get at the protein. And this is similar for Ebola virus. COVID viruses have some protein molecules on the surface, but they don't obscure the active sites, the spike protein and the other surface molecules. So anyway, that's kind of an aside, but I thought it was interesting. Okay, so this is where proteins come from. This is a cell. This is the nucleus of the cell. The DNA is in the nucleus. It has the genetic code, 3 nucleotides that unique and on a unique sequence determine what the where each amino acid is going to be in the protein eventually. And they send that message out to the cell by copying the DNA into an RNA molecule, which it exports to the cell. And this is now a messenger RNA molecule. And this is what is in the Moderna and Pfizer vaccines, is a mildly modified messenger RNA molecule. So the messenger RNA molecule finds other RNA molecules in the cell called transfer RNAs. And these are specialized RNAs that can bind an amino acid at one end and have a complementary code at the other end, which enables it to line up along the messenger RNA. And as it lines up, it approximates the amino acids that it's holding, and they are stitched together in a growing chain, and that's going to become a protein. So that's where proteins come from. Is that as clear as it needs to be? I don't hear a yes. All right. So the summary point is proteins, a potential protein.
I said, yeah, I would say yes, but you won't hear me.
Okay. A potential protein molecule. So when we do this, if you can unmute yourself and make a comment if you want to, I think. Okay, so a potential protein molecule has an almost infinite number of ways it can be uniquely specific. So that's going to be important for the vaccine and the immune system. So the next concept is what's called transmembrane signaling. So here's a simplistic representation of a cell. This is the nucleus where the DNA is, and this is what we call the cytoplasm, where a lot of stuff is, but we never draw it in these. And this is the outside. And the cell has to know what it's supposed to do. So a mystery in the 40s and 50s in molecular biology was, or biochemistry at least, I don't think molecular biology was invented yet, was how does this occur? And so how it occurs is the concept of transmembrane signaling. And this is a more accurate depiction of what the surface of a cell looks like. This is a lymphocyte, which is a white blood cell involved in the immune system. And these things sticking out are not individual molecular structures. They are just additional surface area on the cells, and they just create more surface to have a greater statistical impact from the things that they do. And this is a representation of the inside of the cell, just so you can see that it's pretty complex. And most of it I'm not going to talk about, but this cell can export molecules, and it can recognize molecules on its surface, and that's what we're going to talk about next. This is a section of the cell membrane. And so this is on top here is the outside of the membrane, and underneath is the inside of the membrane, inside the cell. And these long molecules are called phospholipids. This is a lipid, which is a fatty acid, basically, with phosphate groups on either end. And you can see in this cartoon that there are structures that are sticking out in the middle of the membrane. And you can probably see where we're going with this. Okay, so this is a cartoon of the cell wall that I showed you, the cell membrane with the phospholipids. And then this is a protein molecule that is what's called a transmembrane protein, because its structure goes across the cell. And the proper transmembrane protein is such that if you attach something to it at this end, it has enough wiggle and flexibility that it can actually wiggle a little at this end. And what wiggles at this end inside the cell then sets up a process that initiates a reaction of whatever the particular protein is supposed to initiate.
It's 7:30.
Okay. And so a transmembrane, so what you have that we're interested in is a transmembrane protein that has a receptor on one end and a signaling domain inside the cell on the other end. And the receptor will bind something that will cause some kind of change at this end inside the cell that will then trigger a bunch of other reactions. It could be that it opens up a binding site for some enzyme to attach a phosphorus to. It's a whole different lecture, but that's the point here. So I'm not going to do more on that. So the concept is receptor on the surface, and something binds to it. And the term ligand is the general term for something that binds to a receptor. And there's a whole class of ligands called cytokines. And cytokines are small molecules that are really signaling. They're like hormones, except the definition of a hormone is it's made in an organ in one part of the body, like the thyroid, and then it goes in through the blood to have an effect on other organs in the body. Cytokines are kind of like local hormones between cells. And there's a huge variety of cytokines and a huge variety of messages that they can send. And when we draw these cartoons, it's kind of like, okay, there's only one receptor, but in fact, there are numerous receptors on the surface of a cell and numerous cytokines that can be bound and numerous responses that can be triggered even at the same time, and they can balance each other or not. So that's the concept. So this ligand binds to this cell and it creates a change inside. I sort of represented it here as if it initiates the cell to commit suicide and die in a process called apoptosis. But the point is receptor binding, transmembrane signaling, and then a consequence because the activation of enzymes inside the cell triggers the DNA to make new enzymes and activate existing ones and perform some manipulations. And in this case, it would be telling the cell to die. So that's transmembrane signaling. And I have to move this again so I can see what I'm. So these are some examples of what can happen. In this case, a receptor or some represented by one binding molecule triggers the cell to do what's called proliferate, which means make a whole bunch of different copies of itself. So if you're the immune system and you have a new antigen, some foreign substance, some evidence in a molecular structure that recognizes an antibody that is on the surface of a cell, then one of the things you want to happen is you want to make more of that cell because it's the cell that recognized the antibody. Do you see what I'm saying in that example? You can nod your head. Okay. All right. In another situation, if you've had an immune response, it's controlled, and you've created a whole bunch of different cells, and you don't need to have them there anymore. They might be making your lymph nodes swollen or whatever. And so the cells that you don't need, you can trigger to die. And there's a receptor on the cell called the death receptor that if it's activated, it can cause the cell to commit suicide. And then another potential response could be that you could tell the cell to differentiate, which means it could transform itself internally into a cell with different properties. And so in the immune system, this happens when a B cell, which produces antibodies, finds a receptor that it is specific for, and it differentiates into what's called a plasma cell, which just becomes a factory for producing more and more of the antibodies. And then any of these circumstances is going to trigger some kind of cytokine response, and there are any number of possible combinations, but cells can make cytokines themselves in addition to respond to cytokines in the milieu that's outside. And so one of the responses would be to make more cytokines. And this is just to illustrate that transmembrane signaling is complex. And I'm not going to try to explain this other than this is a cell, this is the nucleus, these are the genes and they're being regulated. And these are all receptors of different kinds on the outside. And these things going on are very interspersed. And one of the things to realize from this is these are very complex interactions, and they depend on the existence, not only the existence, but the steady state concentration of one of the participants in one of these pathways at any given time. And there are so many ways that each one of us as individuals could differ slightly in one component of this that could affect exactly what happens. And it might not be a way that would be fatal or advantageous. It just might be a difference that would change a receptor, because these receptors that are on the surface can also be in structures inside the cell. But there's a lot of potential for individual variation in the complexity of these interactions. Okay, so that's all I'm going to say about that. Whoops. Okay. So the immune system. So in strictest terms, the immune system includes your skin and your mucous membranes and your stomach acid and the lining of the GI tract and everything that separates your internal body from the outside world. The way your gut is really the stuff in your gut is really in the outside world, and the rest of you is the inside. But we're going to talk about what has been called the cellular immune system, and there are two arms to it: the acquired and the innate immune system. And I just want to one of the white blood cells that I've already mentioned is called a T lymphocyte. Another one's called a B lymphocyte. I may say T cell and B cell, and I mean T lymphocyte or B lymphocyte. The T cells have a specific receptor, as do B cells, and antibodies are, the B cells make antibodies, and their receptor is essentially a copy of the antibody that they make. And then antigens we've talked about, epitopes I'm not going to really talk about, cytokines I've talked about. And what we're getting at here is how does a cell, how does the immune system make a diverse enough repertoire that it really has cells and antibodies that can respond to a huge variety of antigens. And so it's how that repertoire is generated that I'm going to talk about in the next few slides, I think. Okay, so this is a way of classifying the immune system into what's called innate immune system. And this is a system that we are born with. It is programmed into our genes. It's probably active even in late development in utero, and it depends on a specific but finite and relatively small repertoire of patterns that can be recognized as molecular patterns with specificity. And these are called PAMPs and DAMPs, and that's not really important, but I'm going to say what they are. I'm not going to talk about these. I'm going to briefly talk about macrophages and show you a movie. I'll mention complement, but these are all complements of the innate immune system. The innate immune system is the first to recognize an infection. It begins the immune response, and it activates the acquired immune system, which is this here. And it's sort of less regulated, so I'm using this kind of aphorism, like it's a little bit like a bull in a china shop. It can, if it's managed properly, it can do a lot of good. If it's not managed properly, it can do a lot of damage. And then what we acquire after birth is an immune system with a huge repertoire, maybe of the order of 10 to the 8th possible unique species. I don't know what the number is, but it's some very large power of 10. And I think the most useful subdivision here is B lymphocytes, which come from the bone marrow, differentiate into plasma cells, and B cells are where the antibodies come from. T lymphocytes come from a gland called the thymus, which I'll show you in a minute, and they are subclassified crudely here into CD4 and CD8 cells, but that's not important because there are many further classifications. And functional T lymphocytes acquire specificity from the thymus gland, and B lymphocytes require the specificity from the bone marrow. And in contrast to a bull in a china shop, I think I use the aphorism that this is like a laser-guided set of tools. So PAMP means pattern associated molecular patterns, and these have evolved over time among the species. They come from a few species of bacteria. DAMPs are from damaged cells in the body, and these are the molecules that form the entities, the antigens, really, except they're not really antigens because it's not antibodies that they recognize, it's receptors. And so this functioning of the innate immune system is often called pattern recognition, and the receptors are called pattern recognition receptors. That's all I really need to say about that. So this is really the same stuff. Huge repertoire in the acquired immune system, limited repertoire in the innate immune system. And this is just another statement of what antibodies and antigens are. So this is a cartoon representation of the structure of an antibody, which is also a receptor on the surface of a B cell, and also the T cell receptor. And these things, these structures for both of these kinds of white blood cells called lymphocytes have a constant region, meaning they're true for every possible antibody that you could make. And also on the T cell receptor, a constant on every receptor. But they have this external part that's a variable region, and how this variable region is created is the gist of how the repertoire is generated. So these stick out. This is really an antibody, and this is a kind of a representation of a B cell with antibodies on its surface. And when the B cell gets activated, it makes more antibodies that go out into the circulation. This is the T cell, and the T cell acts by something, another cell comes and sticks an antigen in this T cell receptor because the cell that's doing this was actually infected and it got the antigen from inside its cell. And so the T cell knows that the cell that stuck this to it is infected and it kills it. So T cells kill infected cells, and antibodies bind infected cells and activate other parts of the immune system by cytokines, but also by the complement system, which I'll describe briefly in a minute. Okay, so these are a couple things that I'm not going to talk about very much, but just to mention. Tolerance refers to how your immune system decides that it doesn't want to attack your own molecules and differentiate your own molecules from foreign molecules. And it's a very important concept, but I'm only going to talk about it a little bit. And activation is just a name that many of the cells in the immune system are in a resting state where many of the processes that they need to be functional are not activated. And so when the immune system is activated, it means that they are activated. And transmembrane signaling is what initiates signals that turn on genes that begin to produce the products needed to implement the cell's immune functions. So generating a diverse immune response and a diverse repertoire. So the thymus is a gland that's located in the chest, high up in the chest. It's not the thyroid gland, which is located up here. It's lower down. And this to me is what happens in the thymus is to me one of the most phenomenal things in molecular biology. So there are two things that occur, and one of them is that cells that enter the thymus undergo rearrangements of the genes coding for the variable regions of their receptor. We're talking now about T cells, so it's the T cell surface receptor. So the rearrangement of the genes now creates a whole bunch of cells that have coded for a variable, I guess, protein structure in the part of their structure that I showed you that's the variable region. And on these structures, it's like these parts and these parts and these parts, and then there's a constant region. And the constant region, among other things, binds a molecule called complement, which I'll talk about later. So now you've got a whole bunch of different genes for the variable region. So you've got a whole bunch of T cells that are identical in every way, except that the variable region of their receptor is different for each of the T cells. And so each T cell can form a clone. And so you have now a diverse repertoire of clones, and maybe as many as 10 to the 8th different unique clones. Now, partly related to the concept of tolerance, there's a second function that happens in the thymus, and that is selection. And it's not only because of tolerance, it's because of effectiveness of the gland. And this is, so the selection from among these variable regions occurs because these cells in the thymus the receptors on their surface are exposed to entities that bind to them that simulate the entities that are in the individual. Let me back up and say that, because this is really important. There's a bunch of structures in the thymus that mimic the structures in a normal cell and in a normal body. And in order to get rid of the members of this repertoire that have been created that would potentially attack their own structures, the binding of these structures in the thymus sends a signal inside the cell to die. So some of these cells die because they are potentially autoimmune. Some of them die because they bind so tightly that they really can't function effectively. And those are the ones that could be potentially autoimmune. If they don't bind at all, then they probably aren't going to be very functional as immune components, and they die. So you have a process that goes on. One is the rearrangement of the genes, and then selection for the members that were selected, that were created of individual protein structures that you don't want, and you are left with a repertoire that works. And so, again, these are just cartoon representation. So here's just a recapitulation of what I just said. This is the thymus. These are precursor cells, come from the bone marrow. They go to the thymus. The genes that code for the variable part of the T cell receptor undergo rearrangements so that they now code for a whole bunch of different receptors. And then the ones that are not desirable are selected out by what are called positive and negative selection. And then what emerges is a set of T cells, each one of which has a unique receptor and can form the basis of a clone for that particular receptor with that particular set of specificities. And then when an antigen occurs, like something related to an infection or whatever, it's taken up by certain cells that are called antigen presenting cells, and they interact with a T cell receptor and present the antigen by, often this is an antigen that's generated from inside this cell, because if it's an infection, this cell has become infected. And so it puts the antigens on the surface, presents them to a C cell, and that sends messages to activate the T cell. The T cell proliferates, makes more T cells. They attack the infection. And then when the infection has been controlled, there are signals that are important and tell it to deactivate. And some of those cells will persist as memory cells, and they will be preferentially activated if the same antigen reappears. And so a similar process occurs in B cells, except that it happens instead of the thymus, it happens in the bone marrow. And what emerges is a bunch of B cells that have an antibody on their surface. And when they encounter an antigen, they proliferate, and they undergo a further set of maturation in which they do in lymph nodes. And this is called affinity maturation. It's where the particular B cells that seem to have bound antibody effectively are further rearranged into genes that select for more and more effective antibodies. And so you end up with a diverse collection of antibody producing cells that's further made more specific. I hope some of that's clear. Okay, and just to show you that you can make this much more complex, something that's important is that in T cells, the repertoire depends on the thymus, and the thymus deteriorates with age. When I was in medical school, it was taught that the thymus is pretty much inactive by the time you're 20 years old. That's probably not quite true, but it certainly deteriorates with age. So if something deep, you have a repertoire of T cells and something diminishes the number of those in the repertoire, they may be diminished in a way that selectively diminishes them equally, or that selectively diminishes them unequally, so that you have a residual repertoire that's not very diverse anymore. And then when you reconstitute, either because something goes away or whatever, if you reconstitute from a diverse residual repertoire, you come out pretty good. But if you constitute from a repertoire, a residual repertoire that's not very diverse, then you can generate a lot more T cells in terms of the numbers, but you can't generate the diversity without the thymus. So that's one of the reasons our immune system gets worse with age. And in the specific case of HIV, HIV attacks T cells and causes this diminishment of the cell numbers. So you can reconstitute, but you don't get the same result. Okay, so the rest of what I have to say is just how the immune system does what it does once it's working and identifying cells. So it identifies bacteria by and abnormal cancer cells. It may phagocytose, which means it just eats up certain cells in the immune system. Macrophages in particular just eats up bacteria. I'll show you a movie of that. And then it identifies infected cells, and they're presented to T lymphocytes and killed, or they're represented by pattern recognitions. And then cytokines promote the functions that do all the things that I've talked about. Complement cascade is activated. Cytotoxic granules are one way cells are killed, and then activating the cell suicide pathway is another way. So this is a way CD8 T cells kill. So this is the T cell. This is an infected cell. No, I'm sorry. This is the infected cell. This is the T cell. And this cell has identified a portion of this virus internally, presented it to a T cell, and caused the T cell to synthesize and emit cytokines and toxic granules, which can perforate the infected cell and kill it. And it can also activate this receptor, which is supposed to be the fast receptor, which is what's called the death receptor, and trigger cell suicide. Okay. These are cells and you can look at this, but it's not real important beyond what I've said. This is a cartoon of the innate immune system. Just again showing that this is a very complex interaction. It involves a lot of different kind of cytokines and feedback and regulation. And that's just the whole point of the slide. And this is another example of that. And another example. And complement is a way that antibodies kill cells. Antibodies attach to a structure that their variable regions are specific for, and the constant part of the antibody attaches a fragment of this cascade of proteins called complement, and then this triggers what's called the complement cascade. And I was always frustrated that I could never memorize it when I was in medical school. But it ends up creating molecules that bind ultimately to this cell here and kill it by forming a killing complex at the surface.
Oh, gobbled it up.
Okay, well, I think that's pretty cool anyway. Okay, so I think this may be the last slide. And the point of this slide is just that, well, it's not the last slide, but it's getting close. There has to be feedback. And one of the problems with immune responses, if it's not properly inhibited when it should be by feedback, then you get a phenomenon called a cytokine storm, where things are happening out of control, cells are being destroyed, and inflammation is occurring when it shouldn't be. And that's what this diagram will. And so just to bring it back to COVID, these are informational and you can look at them, but the point is, here's a typical case of COVID. It's just time along the right. And so this is viral replication. Viral replication occurs and somewhere inflammation, whoops, somewhere inflammation occurs, and at its early point you could administer antiviral therapy directed at the virus. But by this time, what you're really concerned about is the inflammation. And if your immune system works the way it should, it will diminish that inflammation and you'll feel much better. But if it doesn't, you'll have an inflammatory response, and that's one of the potential bad occurrences, because once you get an inflammatory response, it can affect every organ in the body, can be ineffective in controlling the virus, and it can affect your lungs and your liver, and that's why people die. And I think this slide, this is a normal progression. This is the innate immune system's activated, then it activates the acquired immune system that brings out T cells and antibodies. The viral load goes up initially and then goes down. But if it doesn't diminish properly, then the innate immune system continues unabated and the viral load continues. And although you have the antibodies and the T cells, you're just out of control. And this is a cartoon of what's called a cytokine storm, meaning inflammation is going on and it's not being inhibited. And all of these blue rectangles are drugs that have been proposed as ways to diminish the immune response. There's been variable success in this. And this is just a slide of my version of my definition of science that isn't really part of the presentation. So I'm done.
Well,
almost.
I am struck by the absolute complexity of this. I'm wondering, how do scientists figure out these things? Half of your slides show pathways of signals going from one place to another, maybe going into the cell. But how do scientists figure this out when there's dozens of pathways occurring at the same time? How do they know that they've got the right one when they try to draw a diagram?
Well, the sophistication of the tools that can be employed has really just burgeoned, because now you can study individual cells in culture. You can isolate components of the cells and look at them individually outside of a cell. You can do genetic engineering to knock out either in a cell itself or even in an animal like a mouse, what's called a knockout mouse. You can knock out a gene and see what happens. But I don't know. You may not want to see any more of me. It occurs to me that I could try to summarize some of the tools.
Go back to slide number 22, for example, that shows the multiple transmembrane signal pathways. And I see in there a whole lot of arrows and lines that go back and forth, mainly from the cell wall into the nucleus.
Is this it?
Number 22. No, it's the cartoon of multiple transmembrane signaling pathways.
Oh, okay. Yeah, this one?
Yeah, that's the one. Now, I see a lot of arrows here showing one thing follows from another. And I just don't understand how it's possible for them to figure this all out.
Well, it is amazing what's been figured out, and also it's humbling what hasn't been figured out, because even if you know all these components, you don't really know the rates at which each of these steps occurs, and you don't know the steady state concentrations. And I don't know if anybody can ever do that, but you can tease out a lot of these things so there's an old joke when I was in graduate school, I was interested in enzyme mechanism. How do enzymes catalyze reactions? And there was a lot that wasn't known then about it. And I worked with a man named William Jenks, and he was probably one of the premier people, but he was a very humbling guy, and he didn't blow his own horn very much. So he, I think, didn't get the recognition he deserved. But he worked with what were called model systems. He, instead of using a big enzyme, he'd look at how small molecules interacted with each other and make models of what was really happening in an enzyme. And he had this joke that he would always tell about why he did this, which was about a drunk who is, a guy comes along and he sees a drunk under a lamppost looking for something. And the guy says, "Can I help you find something?" And he says, "Sure." And he says, "What is it?" He says, "My wallet." And so the guy says, "Well, where is it?" He says, "It's over there in the curb. That's where I lost it." And he says, "Well, why are you looking here?" He says, "Because there's more light." And
Yeah.
So that's what Jenks always said about why he worked with model reactions. But it's fascinating how I listen to a seminar each week by graduate students working in immunology, each of them on different systems, about how they're doing the experiments they're doing.
Well, while we're on this slide, you were talking about signaling from the outside to the inside, and you showed a cartoon of something on the outside wiggling around and having an effect inside. Basically, this has a receptor on the outside. Does it take an activation of the receptor by something plugging into it in order to
Yes.
have the activity
Yes.
inside occur?
Yeah, that's what I was trying to. That's a fundamental part of the process.
So without anything activating the receptor or plugging into it, those effects inside are benign. But when something...
Well, may not even be going on. Some of those effects are generated by what the genes are that's being made, and also what the receptor molecules are being made. Receptors are another thing that can be sent out to beyond the surface if there's a need for more of receptors of certain kinds. So you don't always have all of the receptors there at once. Although you do in this slide, you have receptors for many of the processes that are not even just concerned with immune activation.
Well, most of the receptors are good things, communication between your body and the internals of the cell to have it do something good. But once in a while, something bad comes along. Can a good receptor, something does something good, turn into a bad one? Or are the bad ones just sitting there waiting for the right virus?
You don't really, you could have a mutation that would make a receptor, sensitive to a ligand when it wasn't, and that could be bad. But that's an unusual event. Probably most often, if that happens, it happens at an early enough stage in embryogenesis that it doesn't develop fully.
Any other questions? Have to unmute.
Okay, I'm sorry. I hope that some of that was useful.
Thank you. Yeah, well, that shows a lot more.
About
this.
Did you at least understand the general concepts that you have to bind to a receptor to make something happen, and the way you generate the diversity is by rearranging the genes for the receptors that are involved in antibodies and T-cell receptors? I don't know. Look at the slides and see if anything becomes more clear.
I think there were
Um,
two things that came through. One of them was in the early part, you were talking about how
It's 8:15.
these things were formed, and then how they communicated internally to the cell to do their good things and their bad things.
uh-huh.
And I just, at that point, I realized how complex this was. And that really hit me hard.
Well, I can tell you it's been amazing to follow the progress that's been made over the years compared to when I was in college, what was really known about this, which I thought at the time was pretty advanced. When I was in college in my junior year, I was taking a genetics class, and the lecturer came out and announced that they had just cracked the genetic code. They had figured out which nucleotides code for which amino acids. And then I can tell you maybe 15 years ago, when people had sequenced DNA in an organism and even in a human, they were talking about how most of the DNA was junk DNA. Do you remember those kind of
Yes.
statements?
That's right.
And it turns out that they were full of shit. They said that because they didn't. So when in the genome, the code for a protein has a specific codon where it starts and a specific codon where it stops. So if you know the whole sequence, you can guess where the proteins are coming from by the stop and the start codons. And so a lot of the DNA didn't have obvious stop and start codons, but it turns out that a lot of them are making RNA molecules which actually have regulatory functions throughout the cell that were totally unrecognized, and that it has, I guess, generated a whole new concept, which people sort of knew, but they didn't really know the extent of it, called epigenetics, which is how things can be changed even after you, even after the messenger RNA is generated. So the naive thinking when I was in college was, if we could only identify the genes, then we could figure out what causes all the diseases. But it turns out that, first of all, there are often multiple genes. And I mean, there are a few genes like sickle cell anemia where you can identify one gene and really do something about it. But in many genes, it's turned out that if you take all the people who have a particular disease and you look for a particular sequence of the genome that is common to all of them, then you find that you have maybe 10 different genes and none of them seem to be dominant. And in terms of what you know those genes do, they don't really tell you anything about exactly what happens. So it's a lot more. It's a lot more than just knowing the sequence of the gene of the DNA.
Well, thank you very much, Pete, for this fine presentation about the immune system. It scares me sometimes to.
Well, I think one of the things it illustrates is, why does somebody who gets a particular infection like COVID or lupus or whatever, why does one person have a really bad outcome and another person with the same disease like lupus have a much milder outcome? And you can see all the components of what's happening, that there are any number of places where just a few differences in a polymorphism in the DNA sequence would affect a receptor or an enzyme that was involved in one of the processes and would change the outcome. And so you can talk, it's great to talk about personalized medicine, which is kind of a mantra right now, but you know, we always talked about personalized medicine. From the very beginning, we always said, you don't want to do the same thing for every patient. We've just learned refined differences in how to tell one patient is different from another. But I think the fantasies of some of the people, like presidents who said you could do this with what was the kind of plan they wanted, a Marshall Plan or something like that. We're just very naive about how complex it all is. And you can't personalize us in enough ways to really be able to pin it down in the way that we wish we could. And maybe someday it'll be even better. It's a lot better now than it used to be.